Hello, I'm Sophie Rovner from the American Chemical Society. Welcome to this news briefing from the ACS Fall 2019 National Meeting in San Diego. We're joined today by Dr. M. Reza Qadiri from the Scripps Research Institute. He's studying the remodeling of unhealthful gut microbiomes to fight disease. Dr. Qadiri.
Thanks. Lovely to be here.
So if you could go ahead and give us a little thumbnail sketch of your research.
Sure. Research is about, we understand now that our gut microbiome is like an organ in our body, and its health and its proper functioning is so important, to maintenance of our health, and its dysfunction leads to many diseases, non-communicable diseases, including heart disease, diabetes, and many CNS disorders. So the question has been, because of our Western lifestyle, often the gut microbiome presumably loses its equilibrium, and what we call begins to dysfunction that leads to various diseases. So my research is about how we can remodel a gut microbiome inside a living system, and we have only worked with animal models. And whether the remodeling of a gut microbiome into a functional state would prevent the onset and progression of atherosclerosis, heart disease. And we have shown that we can design various systems, chemicals, and also assays and approaches, and it actually works quite well when given daily oral dose into appropriate animal models of atherosclerosis, that it prevents the progression of it and reduces cholesterol, plaque development in the arteries, etc.
All right, thank you. So we now invite your questions. If you have one, please state your name and affiliation.
I'm Laura Cassidy with ACS. So how do you determine what the healthy or the optimal microbiome is, and that you can then try to correct the unhealthful one too?
Yeah, that's a very good question. That's one of the things that we really don't know yet, because while we might be grouped in a state, we are different a little bit from each other. So in our case, we use the animal model to, I should say our studies will helpfully answer that question in the future. But so what we did, we used an animal model which has been used extensively for development of all the statins that are on the market. So these mice, which are LDL receptor knockout mice, they're perfectly healthy and happy when they're given low-fat diet, normal chow diet. But if they're on a Western diet, which is average of what we consume, over a period of 10 weeks, they develop plaques in their arteries, and they have a high plasma level. So we assumed, based on what is known, that when the same animals, genetically identical animals, when the diet is changed, the gut microbiome is also changed. And it is true, whatever we put in our mouth is a selective pressure, basically, for the gut microbiome. So then we said that if the gut microbiome is causative in development and onset of disease in animals that are on Western diet, therefore that state of the gut microbiome on the Western diet is dysfunctional. And the one on the low-fat diet is obviously functional because they don't develop atherosclerosis. So we chose these two states as our initial point to remodel the gut microbiome of the mice that are on Western diet while still eating Western diet to look like it is on a low-fat diet. Okay, so.
Okay, and how did you identify these peptides that can remodel the microbiome in this way?
Right, this goes back to years of studies that we had done for unrelated projects using these self-assembling cyclic D-alpha peptides that form peptide nanotubes, for which there has been lots of things written about before. And these peptides have an ability to partition in various biological membranes. In this particular case, we're interested in bacterial membranes that live in our gut. And they can modulate their growth profile selectively. So we're talking about hundreds of bacteria that you want to selectively modulate certain portion of the population such that others can grow and fill the niche to look like they're on a, low-fat diet state. So these peptides operate at the supramolecular level. It's a complicated story to describe. So we knew they have a possibility that they can serve as a selective bacterial growth modulators, not killing, right? You don't want to destroy bacteria in the gut. But how do you select for something like that? So we developed an in vitro assay that you're using the entire gut microbiome all at once. And we follow the effect of the peptide, each peptide on the gut microbiome by sequencing the whole thing, and then doing bioinformatics and so forth to choose what we want.
Katie Cottingham from the American Chemical Society. So you mentioned that you didn't want to kill the bacteria in the gut. Why is that?
Well, these are, contrary to what some may think, we are symbiotes, okay? So we have co-evolved with the bacteria that live in and on us. It's not like what I was taught when I was young, that we have developed all these immune system presumably to keep all these guys in checks and balances. So they make a couple of vitamins for us, and that's about it. And actually, we are symbiotes. We have co-evolved. We cannot live without them. Okay, a child born without a functional gut microbiome within the first 3 years will not develop a competent immune system rest of his or her life. Okay, so I consider, and many do also, that the gut microbiome is our 11th organ system. By any definition, it's just like another organ in your body. So the point is not to want to destroy the gut microbiome or kill. Rather, you want to modulate it. In fact, broad spectrum antibiotic use, we take antibiotics for some pathogenic infections, and that often can lead potentially to decimation of your gut microbiome. So no, definitely we don't want to kill anyone, any of these. These are good bacteria.
Okay, and what's your take on fecal supplements, which have grown in popularity in order to...
Fecal supplements?
Yeah, in order to
you want
change...
fecal transplant?
Oh, or that, yeah.
Right.
I've heard of both, yeah.
Well, science has a spectrum, and there are group, and everything has its place. There are a group of patients, IBD patients, etc., that they really almost have no gut microbiome, or is what we call its biodiversity is so drastically changed, right? So I believe that can be quite useful, okay, to transplant microbes, okay, into patients that really can benefit from, because they don't have any. What I'm talking about is the rest of us. We have a gut microbiome that might be dysfunctioning, okay, to correct that. I think it will, science will go forward not using fecal transplant rather than consortium of highly purified and controlled microbial treatment. Okay, so that has its place. It's a very useful thing. Yes.
Okay, so maybe someday doctors would prescribe a pill that would contain your peptides, perhaps, instead of or in combination with the fecal transplant.
No, that won't happen. No combination with fecal transplant.
Other questions? All right, well, thank you very much. The archived version of this session will soon be posted at bit.ly/ACS 2019 San Diego. Please join us for our next press conference at 2:30 p.m. today on a skin patch that could painlessly deliver vaccines and cancer medications in 1 minute. Thank you.